Hi class, let's continue our discussion of chemistry. This chapter is a brief overview chapter in the field of chemistry. So let's keep going. We're going to next talk about organic molecules and how we can make them and how we can destroy them. The process of putting together a organic molecule typically is going to be known as a dehydration reaction. During this process, we take two separate subunits, two separate monomers, and connect them together. And as we connect these two separate subunits together to make a bigger molecule, we're going to have a waste water molecule. Water is a waste product, and as these two molecules are connected together, water will be produced and excreted. And if we want to break down these larger molecules, we actually are going to need to add a water molecule. A water molecule is going to be required to break apart a larger organic molecule and break it into smaller organic molecules. So here we can see a dehydration reaction. One subunit, one monomer has an OH, the other monomer has the H, and they donate those two together to form a water molecule, and a new covalent bond holds the two subunits together. If we want to break those subunits apart, we need to add a water molecule to restore the OH and the H that were removed. If we look at all these molecules that we have in our bodies, these organic molecules, it can be broken up into four big categories: carbs, lipids, proteins, and nucleic acids. And these are all going to be carbon-containing molecules. Oftentimes these are referred to as the macromolecules, the four macromolecules or four categories of biomolecules. As we look at these biomolecules, we can see carbs are typically going to be used to have immediate energy use and are going to be based off of a glucose monomer. Lipids are typically going to be for long-term energy storage and to make to make cell membranes. Proteins are for structural purposes. They are made of amino acids and they help to regulate our metabolism. Nucleic acids, or our DNA and RNA, are used to transmit and store genetic information. So, class, concept checked. Does chemically digesting food, breaking it down into smaller pieces, release or require water molecules? So go ahead and pause the video, check your PowerPoint slides, whatever you need to do. Get me an answer. 1, 2, 3, 4, 5. The correct answer is B, require water molecules. If we're breaking down our food, we're breaking it into smaller pieces. And as we break that food into smaller pieces, we're going to be using up water molecules. So the correct answer was B, require water. Let's continue on and dive into a discussion on carbohydrates. A simple carb, or a small subunit of a carbohydrate, is called a monosaccharide. Saccharide is an old school term that means sugar. Mono is a prefix meaning one. All monosaccharides and all carbohydrates in general are made of carbons, hydrogens, and oxygens. For every one carbon atom, there are going to be 2 hydrogen atoms and 1 oxygen atom. Hence the name carbohydrate. The 2 hydrogens and 1 oxygen were thought to be 1 water molecule for every carbon. And it's later been found that no, that's not necessarily the case, but the name has still stuck. We can use carbs for short-term energy use and for long-term, or I would really say medium-term energy storage. For long-term energy storage, we typically are going to focus on lipids. So think of body fat for long-term energy storage. But we can use it for short-term, think of like glucose and blood sugar, or medium-term energy storage. In that case, that would be glycogen in our livers. We can find carbohydrates in a simple form and a complex form. Simple carbohydrates are 1 subunit carbs or 2 subunit carbs. They are both going to be sweet tasting. A classic monosaccharide is glucose or fructose. Those are 2 very common monosaccharides or simple sugars. Another simple sugar could be maltose or sucrose. Sucrose being a 2 subunit or disaccharide sugar. Sucrose is also known as table sugar. That's the stuff that you buy at the store, cane sugar. We can also have a complex carbohydrate. These complex carbohydrates are going to have many many sugar subunits linked together. Sometimes they're branched, sometimes they're unbranched. Different organisms have different kinds of complex carbohydrates. A generic name for a complex carbohydrate is a polysaccharide. It's made of many carbon rings or many sugar subunits. In animals, we can use glycogen to store energy for that medium term or length of time, and it will store that complex carbohydrate in our livers. In plants, they will have energy storage in the form of starch, and we as human beings enjoy eating starchy foods because they have lots of carbs that contain energy that we can use. So, class, concept check. If we were to eat some cake, which of the following carbohydrates would we likely not be eating, or would likely not be consumed? So think of a cake. It has its frosting, it has its bread. Would we not be eating glucose, sucrose, glycogen, or starch? So let's think about these carbohydrates. Glucose is a simple sugar. It's very sweet tasting. Sucrose is also a simple sugar that's very sweet tasting. Those two sugars are probably going to be present in the frosting and the bread itself. If we look at starch, starch is a complex carbohydrate present in plants that forms the main body of the bread of the cake. Glycogen is a complex carbohydrate that is stored in animal livers. And hopefully your cake does not have animal liver in it, so likely you would not be consuming C, glycogen. Let's move on. What are lipids? Lipids are any biomolecule that does not dissolve in water. It is hydrophobic, water-fearing. It does not like to mix with water. As we look at our lipids, they are oftentimes going to be used as either energy molecules or to make cell membranes, or as steroids. We can also call our lipid a fat or an oil. Generically speaking, a fat is a lipid, an oil is a lipid, a steroid-based hormone is a lipid. We have lots of different kinds of molecules that don't like to mix with water. All of these molecules are lipids, including phospholipids. So let's talk about fat versus oil. Fats, generally speaking, come from animals, whereas oils, generally speaking, are going to come from plants. At room temperature, most fats are solid and most oils are liquidy. In animals, we can use fat for very long-term energy storage. We can also use these fats, or triglycerides, as they're sometimes referred to as, to help cushion us and protect us. We can also use these fats to help insulate us, to help us maintain a constant internal body temperature. If we look at the generic structure of a generic fat molecule is going to have one glycerol and 3 fatty acids. And as we break down this fat molecule, this triglyceride, it's going to turn into 3 fatty acids and 1 glycerol molecule. And this breaking down of the triglyceride requires the input of water. We have the dehydration reactions and then the condensation synthesis. Or hydrolysis, I should say. Hydrolysis, where we use water to break apart the chemical bonds that hold the fats together. Sorry, I was mixing and matching my terms inappropriately there. My bad. We can also understand fats when we look at the nutrition labels. Generally speaking, the average recommended dietary intake is 2,000 calories a day. And this should really be calories with a capital C. And if we're looking at that, the amount of fat recommended for that diet is 65 grams of fat. Of that 65 grams of fat, let's figure out how many calories that would be. So fat is 9 calories per gram, 9 times, I'm using a calculator right now, 65 gives us 585 calories every day from fat is the recommended max. An important thing to know when reading the nutrition information is how many servings we're looking at. The percent daily value of 5% less is low, and a percent daily value of 20% or more is too high. Generally speaking, you want to try to stay away from trans fats. Trans fats are a special kind of unsaturated fat where the hydrogen atoms are located on opposite sides of a double carbon bond. And we want to try to stay away from these trans fats because these trans fats, generally speaking, take more energy to break down. They aren't produced naturally in nature, and since they are more chemically stable, they're harder for our bodies to process compared to the opposite, the cis fats. So let's look at a following nutrition label. Would you want to eat it? Why or why not? Oh, and that nutrition label is right here. So let's skip to our concept check question. Would you want to eat the food on this nutrition label? Why or why not? I would say, you could say, yes, it looks delicious, because, oh my gosh, look at that. It's about half fat, gall darn it. And, hey, look at this. It also has lots of sugar present in it. Or no, it'll taste bad because I don't know, maybe you're someone who doesn't like to eat fat and sugar. Or you could say, yes, it has many vitamins and minerals in it. Or you could say, no, it is very high in fat, particularly saturated fat at 30% of their daily value, and trans fat, 15% of our daily value. So if we look at this nutrition information, it has lots of the stuff that we're trying to limit, and it does not have a lot of, it has no fiber, it has little protein, and has very few vitamins and minerals in it. So in terms from a dietary perspective, you would say, no, it is high in fat, and also no, it's lacking vitamins and minerals and insoluble dietary fiber. So you, generally speaking, would not want to eat this food item. If we look at a phospholipid, another kind of fat, the phospholipid is very similar to a triglyceride, except that instead of having 3 fatty acids, it has 2 fatty acids. Replacing one of the fatty acids, we have a phosphate group with an amine attached to it. And this phosphate group forms the polar head, and then we have 2 nonpolar fatty acid tails. These nonpolar fatty acid tails do not like to mix with water. They do not like being exposed to water, and will preferentially huddle together to stay away from the water molecules that are inside of the cell and outside of the cell. The polar head, though, likes mixing with polar water molecules, so the polar heads can be exposed to the inside of the cell and the outside of the cell. Because of this dual nature of the phospholipids, the polar and nonpolar regions of the phospholipid is capable of spontaneously forming membranes, and is used to form the membranes on the outside of our cells and the inside of our cells. And then finally we have steroids. So steroids are another kind of lipid that are based on 5 and 6 carbon rings. The simplest steroid is cholesterol. Cholesterol is a building block that's used to make both testosterone and estrogen. These steroids are capable of having very profound effects in human beings, even at very low concentrations. It's also worth emphasizing that because these steroids are lipids, they do not like to mix with water. So to transport these steroid-based hormones through the bloodstream, we're going to need special protein carriers to help move them through our watery bloodstreams. And we already covered this concept check question. And to review it again briefly, you know you don't want to eat it because it has lots of fat and not a lot of vitamins and minerals. So let's look at proteins. Moving on to proteins. Proteins are the most numerous biomolecule of the cell. If we removed all of the water from our bodies, most of what is left over would be a protein molecule. The building block, or the subunit, the monomer that we make proteins out of, is called an amino acid. We as human beings have exactly 20 different amino acids that we use to make the proteins in our bodies. Now, other organisms can have more or less amino acids, and it's also worth emphasizing that in a laboratory setting you can create an unlimited number of amino acids. But we as human beings have only 20 amino acids in the proteins of our bodies. What do we use these proteins for that are made out of amino acids? We can use these proteins to act as a hormone. Hormone is going to transmit chemical information to the body. They can make enzymes to regulate the chemical reactions in our body out of proteins. We can make antibodies out of proteins to regulate our immune system, and we can also use proteins to transport chemicals throughout our body. An important aspect of proteins is that they can denature, and when you denature a protein, you change the shape of that protein. If you change the shape, you change the structure, and changing the structure changes the function. Structure is always going to determine function. So if you heat up a protein really hot, that's one thing you can do to denature it. Strong chemicals, strong acids, strong bases are all things that can denature proteins. And this is one of the reasons why cooking our food sterilizes our food. When we cook our food, the high temperature of cooking our food denatures the proteins that keep the bacteria alive, and we start to kill all the bacteria or microorganisms within our food. If we were to look at our amino acids, this is a small sampling. This is only 6 of the 20 that are present in our bodies. All amino acids are going to have a carboxylic acid group and an amino group, but then there's the R group. This R group is a variable group. Sometimes it's a nonpolar group, sometimes it's a polarized ionized group, and other times it can contain sulfur atoms, sometimes it contains carboxylic acids, sometimes it contains nonpolar rings. Each amino acid is going to be different from the next amino acid and have very different chemical properties. The 20 different amino acids with their 20 slightly different chemical properties determines what kind of proteins we make. The amino acids we put into the proteins determine the properties of those proteins. As we're forming a protein, there's going to be 4 primary levels of organization. The very first level, the primary level, is just the limp spaghetti noodle, as I like to think of it, level of organization. This limp spaghetti noodle is just going to be based on the sequence of amino acids, like beads on a string, and this is called the primary structure. And after our primary structure, these polar and nonpolar, ionized and non-ionized amino acids will sometimes form loop-de-loops with themselves. If they form a loop-de-loop, we call it an alpha helix. If they zigzag back and forth, we call it a beta-pleated sheet. And these alpha helices and beta-pleated sheets are known as secondary structures. And these secondary structures can fold back and forth on themselves to form a tertiary structure. The tertiary structure will oftentimes have special bonds known as a disulfide bond, helping to anchor it in place. These secondary structures are going to have weak hydrogen bonds anchoring them in place. We can then take and stop right here at the tertiary structure for many proteins. Many protein molecules never make it past the tertiary structure. It's one continuous sequence of amino acids. Some proteins, though, require multiple strands of amino acids being linked together. And in those cases, we take multiple tertiary structures and link them together to form a quaternary structure. And this quaternary structure is going to be made of two or more polypeptides, or strings of amino acids. So, concept check, class. When we cook food, we denature many of the proteins in it. What effect does this have? Does it change the texture and flavor? It makes it easier to digest. It kills microorganisms, or all of the above. Go ahead and think about this, pause the video, and get me an answer. 1, 2, 3, 4, 5. The correct answer is D, all of the above. By changing the proteins in our food, we are going to change the structure and function of the proteins in our food. So we'll change the texture, and by breaking down our food, by cooking it, by changing those proteins, we're going to make our food easier to digest. And as I mentioned earlier in this video, we are also going to kill the microorganisms in our food by denaturing the proteins that keep them alive. Our last biomolecule category is nucleic acid. There are two primary kinds of nucleic acids, DNA and RNA. DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid. These two different kinds of nucleotides are used for long-term information storage. DNA is used for long-term information storage, and then RNA is used for information transfer. Both DNA and RNA are made of nucleotide subunits. And we use this information in the DNA and RNA to make proteins in our cells. The DNA and the RNA contain the blueprints used to engineer proteins. Each individual nucleotide is going to have a phosphate group, a 5-carbon sugar, and then a nitrogen-containing base. And all nucleotides will have a combination of sugars and nitrogen-containing bases and phosphates to give us some variations. Within DNA and RNA, we'll find that adenine, the A subunits, will always bind with the G subunits. And we find that the A subunits will bind to the T subunits. A binds to T. I misspoke there. I'm so sorry about that. A pairs with T, and in DNA, G pairs with C. However, within RNA, uracil replaces thymine. So in RNA, A pairs with U and G pairs with C. If we look at adenine and guanine, they are double-ring purines. So they're going to be a nitrogenous base that contains 2 rings of carbon molecules. Cytosine, thymine, and uracil are single-ring pyrimidines. So those nitrogenous bases are going to contain single-ringed nitrogen-containing compounds. Adenine and thymine are linked with 2 hydrogen bonds in the double helical structure of DNA, and then guanine and cytosine will be linked with 3 hydrogen bonds. And these hydrogen bonds will hold the alpha helix, or the double-stranded helix of DNA, together. And just to emphasize it again, in RNA does not have any thymines, but instead will have a uracil molecule to replace the thymine molecule. So in RNA, G matches with C, and A matches with U. Some other differences between DNA and RNA include the fact that DNA is double-stranded, RNA is single-stranded. DNA has a sugar known as deoxyribose in it, whereas the sugar in RNA is a ribose sugar. And if we look at the names ribose versus deoxyribose, As the name implies, DNA has one less oxygen in its sugar. It's been deoxygenated. It's deoxyribose. Whereas the sugar in RNA, the ribose sugar, will have one more oxygen in it. So, concept checked. Which is more stable and why? RNA, because it has more oxygen. DNA, because it has more nitrogenous bases than RNA, implied there. Or C, RNA, because it's smaller than DNA. Or DNA, because it is double-stranded. So, pause the video, get me an answer. 1, 2, 3, 4, 5. The correct answer is D, DNA, because it is double-stranded. Because DNA is double-stranded, it has more chemical bonds holding it together than RNA, which is single-stranded. So DNA is going to be inherently more stable. And that's all we have on our chapter on chemistry. Hopefully, you are able to have a brief, or I should say, cursory understanding of the field of chemistry as we're diving headlong into the topic of human biology. If you have any questions, please feel free to post them on the discussion board or shoot me an email. Happy studies!